Hello, and welcome to the Chapter 12, Parts 1 and 2 online lecture on the cell cycle, cell division, and mitosis. You should use the information in this lecture to complete the Chapter 12, Parts 1 and 2 guided notes, which of course you should complete before you attend class. There are certain times in the life of a cell when it's necessary to increase their numbers or to decrease their numbers. Cells can increase their numbers through a process known as cell division, and you guys have all heard of cell division before. This is where one cell will divide into two or more daughter cells, and they pass on their genetic instructions to that new cell. Single-celled organisms such as paramecia and bacteria use this to increase the number of cells in their populations. Multicellular organisms use cell division as well. This is useful in healing a wound, for instance, or in growing our bodies. Now, believe it or not, sometimes it's important, especially in multicellular organisms, to decrease the number of cells that exist. We refer to this process as apoptosis, or more dramatically, programmed cell death. Sometimes it's also called cell suicide. In this process, special signaling molecules will bind with special receptors on the membrane, and that will cause the cell to basically implode. So the membrane does something called blubbing, where it gets really leaky and lumpy. The cytoskeleton begins to break down inside, so this thing starts to fall in on itself, and the whole cell will fall into these little vesicles filled with different cell components. So the cell just breaks apart. These little components can be recycled into other cells. Now, why in the world would you ever want your cells to kill themselves? That seems really counterproductive. I mean, the more cells, the better, right? Well, not necessarily. Sometimes it's counterproductive to have too many of a certain type of cell. Sometimes you have too many of a certain type of cell in the wrong place at the wrong time, and you need to get rid of them in order for the body to function properly. Let me give you two examples of this. Now, hopefully you guys got to raise or at least see tadpoles when you were a little kid. You know that baby frogs start out looking a lot more like fish than they do frogs, and at a certain point they lose these tails and grow legs. Where do the tails go? They don't just fall off. Well, what's happening is that the cells inside these tails are going through apoptosis. They are breaking down, they are falling in on themselves, and then the body can reabsorb them and use those materials to grow legs. A similar thing happens in human embryos when we are very tiny. We all start out with these hands that look like little paddles. They look like little paws, and there's skin between the fingers. So we have webbed fingers. At a certain point in development, the webbing between the fingers begins to go through apoptosis. Those cells die, and that actually separates the fingers from one another. And yes, it is true that occasionally a baby will be born with webbing between the fingers, and oftentimes the parents choose to have that corrected surgically. Another important reason that a cell might want to go through apoptosis is if it's damaged beyond repair. Consider this. What if a cell or a group of cells is permanently infected by a virus or a bacterium, and that virus or bacterium is just going to spread to other healthy cells? In that case, it's probably more beneficial to kill off those cells, to sacrifice those few cells to save the rest of the organ. And so that's often what the body will do. This also applies to cancer cells. If the cancer is caught at an early enough stage, sometimes they can be signaled to go through apoptosis and break down instead of continuing to divide. Other times, cancerous cells don't receive that signal or ignore that signal and will continue to divide and grow an even bigger tumor. Before we dig into cell division, there are several important ideas that you really need to understand, and the first of these is the cell cycle. The cell cycle represents the life of one cell. So it's all of the events that occurred during a cell's lifetime, all of the proteins that are made, all of the processes it participates in, all of the events that occur. Now, measuring the life of a cell can be a little tricky. Different kinds of cells live different amounts of time. So how do we do it? Well, scientists define the life cycle of a cell, one cell cycle, to be all of the events that occur from the time the cell is divided until it divides again. So from one division to the next, all the things that the cell goes through. That's the cell cycle. The cell cycle is divided into two major phases, known as interphase and cell division. Cell division, of course, is that period of time when the cell is dividing into two, it's reproducing. At the end of this phase, we say that a new round of the cell cycle has begun. Interphase represents all of the rest of the events of a cell's life, when it's making proteins and doing its job and so on. Now, interphase is another example of something that's poorly named in biology, because the other term for interphase is the resting phase. Why would you call it the resting phase? It's not resting at all. Cells are really, really busy during interphase, doing all of their life stuff. Well, there's a historical context for this. When the very first microscopes were developed, they allowed scientists for the first time to actually look at living cells, and that was very exciting. Cells were especially exciting to observe during division because you could see stuff moving around in there, the cell divides in two and changes shape. Really fun to watch. But the rest of the time, when the cell's not dividing, they're just kind of sitting around not doing anything, according to those early microscopes. So they named it the resting phase. Today, of course, we know that the resting phase is sort of a misnomer, that there is actually a lot of stuff going on inside cells during interphase. But sometimes you still hear that old term being tossed around. Interphase can be divided into three smaller phases. The first of these is known as G1 or gap 1. Now, gap 1 begins at the end of division. So at this point, the cell is going to recover from division. That probably means it's going to need to grow some new organelles that weren't divided up properly, make a lot of new proteins, and kind of increase in size as well, and kind of recover from that process. Now, while the cell is progressing through G1, it's also, of course, going to be doing its job. That can mean different things for different types of cells. Liver cells, for instance, their major job is to help detoxify the blood, whereas a brain cell's major job might be to send communications, to send messages between cells. The second phase of interphase occurs right at the end of gap 1 and is known as the synthesis or S phase. Cells only enter this phase if they intend to divide again, and the reason for this is that it's during the synthesis phase that the cells begin to make their first preparations for division. Synthesis means to build something. So what are they building? Well, during the synthesis phase, a cell is going to copy its DNA, so it's building DNA. This is that process of DNA replication that we talked about back in chapter 16. So in this point, the cell will copy its entire genome and hang on to those extra chromosomes in preparation for eventually dividing and sending one set of that DNA onto one cell and the duplicate set onto another cell. Also during this time, the cell, of course, has to do its job. It can't stop and take a break from its job, even though it's replicating its DNA. I'm sure you remember this, but just to review, this is what DNA replication looks like. I'm sure you remember that it's the enzyme helicase that actually opens up this piece of DNA and makes the replication fork, and that there's a whole host of other enzymes, including DNA polymerases and ligases and others, that actually do the copying. The timing of DNA replication relative to division is also kind of interesting. There's actually a pretty big gap of time between the two processes. Think of it this way. If cell division is scheduled for, let's say, noon on a particular day, DNA replication will happen long before. It will happen at, let's say, midnight the night before. So the cell actually carries double copies of all of its chromosomes for a long time before division occurs. The third phase of interphase is known as G2 or gap 2. This phase is the shortest part of the cell cycle. During G2, the cell is going to make its final preparations for division. So at this point, it's going to build all the machinery that it needs and make sure it has all the proteins and things like centrioles that it needs to actually divide. And of course, during this time, the cell is still doing its job. It can't take a break even during G2. But once it finishes G2, it's going to be all ready to divide. The last phase of the cell cycle is known as the M phase. This is the division phase, the time during which a single cell, called a parent cell, is going to divide into two or more daughter cells, depending on the type of division it's undergoing. The cytoplasm and the organelles get split into two or more parts, approximately evenly, although it's not always exactly evenly. And the genetic material is going to be split into even parts and passed on to each of the daughter cells. Cells need to be able to very carefully control how quickly they go through the cell cycle. In other words, they need to be able to carefully control how quickly and how often they divide. If a cell doesn't divide at all or divides very slowly, it can cause a problem with tissue replacement, with healing. But if a cell divides very quickly in an uncontrolled fashion, it can cause things like this: tumor growth, cancer. Cancer really is a disorder of the cell cycle. The cell is dividing too fast, it's not dividing in a controlled way, and that can cause lots and lots of other problems. The rate at which different kinds of cells go through their cell cycles differs depending on the type of cell you're talking about. Skin cells, for instance, divide very rapidly. Here we have the dermis of the skin, one of the deeper layers of the skin. And these cells down here are going to divide, and that's going to push these other layers of skin up. And the layers of skin on the outside, the epidermis, get sloughed off all the time. You lose those all the time and are constantly replaced. For this reason, skin is very susceptible to developing things like cancers. It's dividing all the time, rapidly anyway, so it's really easy for it to get out of control. On the other hand, other kinds of cells hardly divide at all. Cells like nerve cells rarely divide. In fact, some of the nerve cells that you are born with, you will die with. They live as long as you do. Cells use something known as the cell cycle control system to ensure that they are dividing and going through their cell cycle at an appropriate rate. Now, there's not actually a little clock built into a cell, but what cells actually use are groups of molecules that work together and they regulate the speed of the cell cycle. These molecules rise and fall as the cell goes through its different stages, and they can act on the cell in a number of different ways. Some of them act on the cell by sending signals, some of that cell communication we saw in a previous chapter. Others help control gene expression of key genes that allow cells to go from one phase to the next. The cell cycle control system depends on something called checkpoints. Checkpoints are these moments in time when the cell sort of pauses and takes a look at all of its chemical components, and it makes sure that at that time it has all of the chemical things that it needs to proceed into the next phase of the cell cycle. There is a checkpoint at the end of G1. There's a second one at the end of G2, just before the cell begins to divide, and there's even a checkpoint in the middle of mitosis, or cell division. And at this point, the cell pauses and makes sure that all of the machinery of division is hooked up correctly and that division is proceeding correctly. There are a number of molecules that work together to make up this chemical clock, including things like protein kinases, cyclins, and some molecules called growth factors. The way this works is that these molecules will rise and fall as the cell goes through its different phases. They work sort of like hormones in a woman's monthly cycle. You know how Hormones go up and down at certain points during the cycle, and that causes different physiological events to happen in the woman's body. Well, on a much smaller scale, we have these chemicals that rise and fall at certain times, and at the checkpoints, the cell will take a moment and look at all the different chemicals. And if all of these chemicals don't have their proper levels at that point in time, it will not proceed into the next phase of the cell cycle. Of the checkpoints, the G1 checkpoint is actually the most important. And that's because this is the point at which the cell decides whether it's going to go on and divide and reproduce or not. The G1 checkpoint occurs at the very end of gap 1, the G1 phase, and it's at this point that the cell looks at its chemical levels and kind of makes a decision as to whether it will go on into S and G2 and divide, or whether it's not going to divide. If it's not going to divide, it will be diverted into another phase of the cell cycle known as G0. This G0 phase really is the G0 phase. It's not the go phase, that's a 0. In fact, it's more like the stop phase, because when a cell enters G0, it's still going to do its job, whatever its job is, but it's not going to worry about division. So it's not going to copy its DNA, it's not going to build centrioles or any of that other stuff that happens in the S and G2 phases. Cells can enter and leave the G0 phase as needed. For instance, cells such as muscle and nerve cells rarely divide, so they can go into G0 and just stay there pretty much indefinitely unless something happens and they need to divide. Other cells will come in and out of G0 fairly frequently. However, let's say that you've been on a really nice diet, you've been eating really nice and healthy, and your liver cells have been in G0 just trucking along. They don't really need to repair themselves very much. But let's say you have a really super fun weekend and you drink a lot of alcohol and eat some bad for you foods, and now your liver has to do an extra amount of work. Some of those chemicals, such as alcohol, can damage your liver cells, and so at that point, the cell needs to regenerate those cells. It needs to come out of G0 and get busy and divide to make more liver cells. So at that point, it can come out of G0, blast through that checkpoint, go through S and G2, and divide some more. When that has kind of stabilized, it's able to go back into G0. So G0 is kind of a holding pattern that allows cells to regulate how quickly, how often they divide. The next important thing that you need to understand really well before we discuss division is what a chromosome is. There are a lot of vocabulary terms that start with chromo, and you need to be able to keep them straight. So first, let's just review what we've learned previously. A chromosome, of course, is one long piece of DNA. Chromosomes can look like X's, sometimes they're just straight bars. We'll talk about why they have different shapes later. But for now, you need to know that a chromosome is a long piece of DNA and that it contains shorter sections that we call genes. A gene is a recipe for making one or more related proteins. Different organisms have different numbers of chromosomes. For human beings, the magic number of chromosomes is 46. So we have 46 pieces of DNA in a typical cell, like a skin cell or a heart cell. How many genes do we have? Well, estimates differ, but somewhere between 25,000 and 35,000 genes are found on those 46 chromosomes. Now, collectively, all of the DNA inside a cell is known as its genome. When a cell is in interphase and is not dividing, the chromosomes will be stored in a form known as chromatin. Let's take a look at it. Here we have a chromosome. Sometimes we refer to this long, straight piece of DNA as linear DNA, but it's a chromosome. What the cell will do is it will take this chromosome and it will wrap it around and around these little proteins called histones to form things we talked about before called nucleosomes. Eventually this gets wrapped up into a fiber called a chromatin fiber. Now, why does a cell do this? Well, DNA is long and stretchy like a long thread, and that can get tangled, it can break, it can be hard to work with. So what the cell does is it wraps it around these proteins and it organizes it. This chromatin fiber is much easier to work with than just a big ball of DNA. It helps protect the DNA, but also gives enzymes access to that DNA when they need it. Storing DNA as chromatin also helps the cell physically fit all the DNA inside the nucleus. Think about this. Even though DNA is super thin, there's 6 feet of it in every single cell. So how do you fit 6 feet of something inside a nucleus that's far too small for the eye to see? Well, you wrap it up really, really tightly, and that's what happens with chromatin. Chromatin is fine for everyday use, but when a cell is getting ready to divide, it's going to need to move its DNA around in a very particular, very organized pattern. So to do that, what the cell needs to do is take these chromatin fibers and coil them up on themselves over and over again and package them and get them ready to move. When the DNA is all packed up and ready to move, it makes a structure that looks like this, kind of an X-shaped thing. And we're going to refer to that structure as a visible chromosome. Some sources just call these X-shaped things chromosomes. And they say that chromosomes only exist when the cell is dividing. But of course, the term chromosome can refer to a long stretch of DNA as well as to this packaged form of DNA. So when I refer to this X-shaped thing, I am going to call it a visible chromosome to help keep it straight. Visible chromosomes are called visible because you can actually see them under the microscope. Of course, when the cell isn't dividing, the chromatin is still present inside the cell, but it's really diffuse and hard to see. So you just call it chromatin. But when the chromatin coils up on itself and takes on this X shape, it's really dense, it sucks up a lot of dye, and it's easy to see under the microscope. So that's why they're called visible chromosomes. So to just reiterate that, chromosomes are always present inside a living cell. They are just long pieces of DNA. But chromosomes are only visible inside a living cell while that cell is dividing. Now, there are techniques for making chromosomes coil up into visible chromosomes outside of the living cell. So for instance, if we took a cell and took all the DNA out and we made those chromosomes coil up, they might look like this. And you'll notice that each of these chromosomes doesn't really have that X shape. They're shaped kind of like bars, and we'll talk about why that is in a second. But these have been made to artificially coil up, so they're visible as well. Visible chromosomes are only visible, can only be seen during cell division inside a living cell. But if we could see inside that cell during the rest of the cell cycle, the chromosomes would take on two forms. They might look a little different at different times of the cell cycle. For instance, if we could see inside the cell during G1, for instance, the chromosomes might look like this. We call this shape unduplicated. And what this unduplicated chromosome represents is just one piece of DNA that's been supercoiled up on itself. If we could see inside the cell, we would find 46 of these in a typical cell, such as a skin cell. If we could see inside the cell at the end of the S phase or the G2 phase, and if the chromosomes were coiled up into their visible forms during these times, they might look like this. This shape is referred to as a duplicated chromosome. Now think about what happened during the S phase. During the S phase, the cell copies all of its DNA. That's when DNA replication happens. So each of the chromosomes gets copied. What this form represents is a piece of DNA and its exact duplicate, its identical twin. And these identical twins are held together with a little protein clip called a centromere. Because these guys are identical twins, we call each half of this X-shaped chromosome a sister chromatid. Here we have the unduplicated and duplicated forms of the visible chromosomes side by side. On the left, we have the unduplicated form. This is what the chromosome might look like if we could see it during the G1 phase of the cell cycle, so early on in the cell cycle. During the S phase, the cell goes through DNA replication. So in the S phase, you get an extra copy being made of each of the chromosomes. If we could see the chromosomes from that point on, late in S or in G2, they might look like this, the duplicated form, with each half of this chromosome representing a piece of DNA and its exact duplicate, the sister chromatids. When the cell goes into division, the two halves of these chromosomes, the chromatids, are going to get divided up from one another. So this represents division. And if we could follow those chromosomes into their new cells, we'd see that again during G1 of the next cell cycle, each of them is in the unduplicated form. So again, if we could actually see these chromosomes during the entire cell cycle, they might look like this. We start out in G1 with the chromosomes being unduplicated, and there would be 46 of these guys floating around in a typical human cell. During the S phase, they all get copied. So that would mean there would be 92 of these things floating around inside the cell. In G2, the chromosomes shape would change. They would actually get together and form this X-shaped thing called a duplicated chromosome, with each half representing an exact duplicate of the other half. And then, during division, these two halves would split up. One of the chromosomes would go to one cell, and the other chromosome would go to another cell, and the cell cycle would start over again. Now remember that the chromosomes are only actually visible, they only actually look like this during cell division. The rest of the time, the chromosomes are going to be in the chromatin form. They will be more loosely coiled, so they might look something like that. During replication, each chromosome gets copied, so that would mean there's two pieces of chromatin floating around for each of the original one. And even during G2, they would still be in their chromatin form, so they wouldn't actually be joined up like this. They're only joined up during division.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
Your 46 chromosomes carries all of your 25,000 to 35,000 genes. And we like to think of this as representing one set of DNA. But really, you have two sets of DNA. You have two sets of chromosomes carrying two sets of genes. Why is that? Well, it's because your DNA came from two different sources originally. You inherited 23 chromosomes with genes from your mom, called maternal chromosomes, and you inherited 23 chromosomes from your dad. We call these paternal chromosomes. You can actually see these chromosomes in a karyotype diagram. So in a karyotype, a cell has been taken out of an individual and has been made to coil up artificially and has been laid out in an organized way, so you can actually see the size and shape of the chromosomes. The chromosomes actually exist in pairs. Here, for instance, is pair number 1, and one of these chromosomes came from this person's mom, and one of these chromosomes came from this person's dad. So you actually have two sets of DNA, one set you got from mom, one set you got from dad, and you have two genes that can influence every characteristic. The maternal and paternal chromosomes exist in pairs that we refer to as homologous. Homo in general means the same. So what's the same about these pairs of chromosomes? Well, let's take a look. Here we have chromosome pair number 1, and you can see that within the pairs, the chromosomes have the same size, the same shape, and the same banding pattern. Also, notice that we organize the chromosomes from longest to shortest. So the longest pair we call chromosome pair number 1, and the shortest little pair over here we call pair 22. There's also pair 23, which represents the sex chromosomes. We'll talk about those guys in a minute. Now back to this banding pattern thing. The location of a gene on a chromosome is called its locus. So that's what homologous refers to, is the location of the genes on the chromosomes. Notice that within a pair, the banding patterns are the same. Those bands represent the genes. So what this tells you is that within a pair of chromosomes that are homologous, these chromosomes carry the same genes in the same locations for the same characteristics. They are homologous. Let me give you an example of that. Let's use eye color as an example. And let's say that the gene for eye color is located on this particular chromosome in that particular locus. That means that the chromosome that you got from mom and the chromosome that you got for dad are both going to carry an eye color gene. Now, even though they carry the same genes for the same characteristics because they are homologous, they aren't necessarily identical. Maybe mom gave you the gene for blue eyes, whereas dad gave you the gene for brown eyes. The two chromosomes don't have to carry identical genes, but they do have to carry the same genes in the same places. So they need to have something that codes for a particular characteristic in the same place on those chromosomes, or they're not homologous. They don't function very well. Now, what color eyes will you end up with if you have a blue-eyed gene from one parent and a brown-eyed gene from another parent? Well, we'll talk about that in the next couple of chapters, but it's definitely the interaction between these genes on these different chromosomes that makes you you, and it's because of these interactions that you are different from all of your siblings and from your parents. Before the dawn of the internet, we had these books known as encyclopedias. And if you wanted to know something, you would go and look in the encyclopedia. They're arranged alphabetically by topic. These encyclopedias are kind of like the chromosomes inside a cell. One set of encyclopedias is going to consist of a number of volumes. And since we have 46 chromosomes inside a cell, we'll say that each encyclopedia set has 23 volumes. One of these encyclopedia sets you will have inherited from your mom, and the other you will have inherited from your dad. Now, both of these sets of encyclopedias have to have a volume 1, Volume 1 contains information about things that start with the letter A. Volume 2, for instance, might contain information about things that start with the letter B. So you don't want to skip any of these volumes or you'll be missing some information. But if you have to do a report on something, if your cell has to use the DNA, it can access either set of encyclopedias depending on the patterns of gene expression, depending on how these volumes are turned on or off within the genome. So having sets of homologous chromosomes is like having two complete sets of encyclopedias inside a cell with volumes that match in terms of the topics that they cover, in terms of the genes that they carry. Now I told you that all of your chromosomes exist in homologous pairs, and technically that's not true. Actually, the only pairs of chromosomes that are homologous, that match in terms of the genes that they carry, their size and their shape, are pairs 1 through 22. We refer to these chromosomes as the autosomes. The chromosomes in pair number 23 are not necessarily homologous to one another, at least not in males in the human species. We refer to these non-homologous chromosomes as the sex chromosomes. If you are a female, you inherit two X chromosomes. So here's the big old X chromosome, and you have two of those. If you are a male, on the other hand, you have inherited an X chromosome and a Y chromosome. Now look how different those two chromosomes are from one another. The X chromosome is gigantic. It carries over 1,000 genes, and these genes are necessary for all individuals to have normal body functioning. Everybody's got at least one X chromosome. Females have two. Look at the little Y chromosome. It only carries about 78 or 80 genes, and all of these genes have to do with making the body male. So these two chromosomes are not homologous to one another if you're a male because they differ in their size, their shape, and in the genes that they carry. This slide has to do with where babies come from and also how babies get their gender. And it starts like this. When a man and a woman love each other very much, they might decide to make a baby together. Or they might get together and intentionally or unintentionally fertilize an egg. That can happen. How will that fertilized egg have a gender, and where does it come from? Well, one of the pairs of chromosomes that they will donate to the offspring will be the sex chromosomes. And it might surprise you to learn that it's actually the man, it's the father, that determines the gender of the child. Why is that? Well, think about it. Females have two X chromosomes, so they can only donate X's to the next generation. But men have an X and a Y chromosome, so they can donate either an X or a Y to the next generation. If they donate an X inside that sperm cell, the offspring will be female. So there's a 50% chance of the child being female. If they donate a Y chromosome, then that child has a 50% chance of being male, or the child will be male. There's a 50% chance overall of that child being male. So it's actually the father that determines the gender of the children. Now, I told you earlier in this lecture that human cells have 46 chromosomes, and that is still true for most cells. However, some cells can differ in the number of chromosomes that they carry. So let's talk about that. Let's start with most cells. Most cells in the human system are what we call diploid. Di stands for two. So what this word means is two sets of chromosomes. We can represent this with a number called 2n. And for human cells, we say that the 2n number is the magic number 46. In other words, if we were to take a cell such as a skin cell and crack it open and make a karyotype of it, we would find 46 chromosomes inside that cell. 23 that came from the maternal cells, 23 that came from the paternal source. So diploid, two sets. Now, which cells in the human system are diploid? Well, these cells we refer to as somatic cells, and they're almost every cell type in the human body. It includes skin, heart cells, brain cells, bone cells. All of those basic body cells are somatic cells and are diploid. The only cells in the human system that are not diploid are these cells we call the gametes. Gametes are sex cells, so these include sperm cells and egg cells. Sperm cells and egg cells have half the DNA in them that you need to make a person, so we call them haploid. I want you to think half haploid. That means that if we took an egg cell or a sperm cell and cracked it open and made a karyotype out of its chromosomes, it might look like this. Notice that there are only 23 chromosomes inside this cell instead of the usual 46. There's one representative of each of the chromosome pairs. So there's still a 1, a 2, a 3, all the way down to pair 22, and there's a sex chromosome as well. One representative of each homologous pair. Another way to represent haploid is N, just one set, meaning one N. And we would say that the haploid number for a sperm cell or an egg cell is 23. Here are the karyotypes for a diploid cell and a haploid cell side by side. And when we put them next to each other like this, you can really see the difference in the number of chromosomes. The diploid cell, the somatic cell, is going to have 46. The haploid cell is only going to have 23. Now you might ask yourself, why would the haploid cell only have 23 chromosomes? How can it even function? Well, think about the purpose of a sperm cell or an egg cell. The purpose of those cells is to get together sperm fertilizing egg. So a sperm cell is going to carry its 23 little chromosomes. It's going to meet up with the egg cell, which also has 23 chromosomes. And when they are fertilized, that makes a fertilized egg with a total of 46 chromosomes. That cell can then divide and make all of the organs and other cells, each of which will also carry 46 chromosomes. All right, excellent work. You guys made it to the end of this part of the chapter. There will be one more part to this chapter, and then we'll dig into chapter 13. Keep up the good work, complete the year turns, and bring all of that to class with you so we can complete it.